everybody. Let's get started. Can you hear me? Hopefully, folks back at Clemson and whoever else is tuning in can hear me. I'm Thomas Rainwater. I think most people here probably know me, but if you don't, I'm a research scientist here at the Brook Institute, and I have the extreme pleasure today to introduce our speaker in today's installment of the FEC Graduate Student Seminar Series, Dr. John Bowden. And it's a privilege because he's a good friend of mine. So Dr. Bowden received his BS in chemistry in 2003 and his PhD in 2009 in analytical chemistry, both from the University of Florida. He was a postdoctoral fellow at the Department of Biochemistry and Molecular Biology at St. Louis University from 2009 to 2011, and then an NRC postdoctoral fellow with the National Institute of Standards and Technology, also known as NIST, in Charleston, South Carolina, which is where I met John back in 2011. And he was there from 2011 to 2013. They liked him so much, they actually hired him. So he then became a research chemist for NIST in 2013 and worked at Charleston at the NIST lab until 2018, at which time he then joined the University of Florida as an assistant professor. Last year, he obtained tenure. Congratulations on that. And has published over 140 journal articles, 60 of them focused on PFAS, which we'll talk about today. His research program is focused on employing novel mass spectrometric methods at the chemical-biological interface, incorporating elements of measuring chemicals of emerging concern, analytical toxicology and environmental chemistry and engineering, and exposomics. And just a couple other words. So I met John in 2011, and we worked together on, he and I and other folks, on a variety of projects in South Carolina, Florida, even South Africa. John spearheaded a project in South Africa looking at pansteatitis in crocodiles and fish, and it did an amazing job of leading that project. But he has more to talk about today. So with that, John's talk is exploring the presence of PFAS in both our natural and built environments. John?
Thank you, Thomas, for that great introduction. And thank you all for the opportunity today to talk about my research. Who here has heard of PFAS? Okay, well, that's good. All right, a lot of rooms I go to, people aren't aware of PFAS, and then by the end of the day, they're all freaked out because they didn't know this thing exists, and it's actually something that's going to be very bad. So quickly before I start, I just want to first thank all the students in my lab that did all the amazing work that I'm going to talk about today. Most of the time, the acknowledgement is at the end, and people are already not paying attention, so I want to make sure put the focus on the students who did all this great work. Also want to briefly talk about what my lab does. So my lab is chemistry focused, and we develop novel analytical tools to measure contaminants of emerging concern in the environment. Once those chemicals are released into the environment, we look at their fate and transport. We also look at various ways they go into different food webs. Also,
it's a little bit echo. I'm trying to figure out why. It might be on this laptop. I think that's, yeah.
Okay.
Okay.
Sorry. Also, a lot of these chemicals are used in a lot of everyday products. So once those chemicals are thrown away, what happens to them when they enter the waste stream? And of course, we also do some toxicology as well. In the field of PFAS, we've been working on this for a long time. There are a lot of things that I wanted to talk about today. I actually cut out a lot of slides last night because I want to make this talk not so long. A lot of things we're going to talk about today, I'm going to talk about very briefly. If anybody has interest in talking about them more, I'll be here for a while. Happy to talk to you about them. And there's a lot of other projects that I'll talk about too, in some cases, very shortly. And other ones I'll talk about a little more in detail. So if you have any questions, just please ask. So we've known about PFAS since the 1930s. And incidentally, one of the first major applications of PFAS was the Manhattan Project. So it was used in the construction of the atomic bomb. Several decades later, they become frequently used in a lot of everyday products. It wasn't until the early 2000s that we as a society started asking this question: these chemicals we're using a lot, are they good for us? You know, they're everywhere. Are they having any bad impacts on our health? And sure enough, they are. It wasn't until maybe the last decade or so that the government actually started to impose regulations or start talking about imposing regulations, because a lot of different lawsuits were popping up. People are finding PFAS in drinking water, and we started to realize that these things are in everything that we are exposed to on a daily basis. So we need to make more regulation. These are two of the most common PFAS. We call these legacy PFAS because they've been around for a long time and used in a lot of different things, PFOA and PFOS. But I just want to point out that these are just two of reported over 15,000 PFAS chemicals. So we have our work cut out for us. And why are these chemicals so bad? So the reasons why they're used so frequently in a lot of our products is the same reason why they're bad in the environment. So as a society, we don't like buying products that break down. We want things that last a long time. We want things that don't stain. We're a product of our own bad behavior, right? So we went to the lab, we made these chemicals, and what you may notice here is this carbon-fluorine bond. And these things are the strongest bonds in chemistry. So once we make them, they have a hard time breaking down. They make our products last a long time. But once they get into the environment, they don't break down there as well. So they last a long time in the environment. The other thing you might notice is that they have this duality of chemical properties. So you have this hydrophobic fluorocarbon tail. And it also has this hydrophilic functional group on the head group. And this duality of chemical properties allows them great utility in a lot of different applications. So one of the things they're used as are surfactants. But these properties also are bad when they get into the environment as well because they increase their mobility. So they can get in the atmosphere, they can go through water, different parts of the water column, they bind to sediment. So they have a very active mobility once they get into the environment. You might be aware of some of the products that PFAS are in. This list is constantly growing. Food packaging, clothing, cosmetics, textiles, dental floss, nail polish. The list is constantly growing. Perhaps the most significant source is the use of aqueous film forming foams that put out fires. These are used at military bases and airports. Once they spray these onto fires, then that foam, which has all this PFAS, then gets stuck into the soil, potentially then run off into groundwater and other water sources. Before I finished last night, I just went on Google to see what other things people are measuring PFAS in. Band-Aids is a new one that I just saw recently, and also smartwatches. So they found that PFAS, particularly PFHXA, is lying on the bands of smartwatches to prevent the bands from being exposed to oils and sweat. So they're everywhere and almost in everything. This is one of my favorite slides because it shows the potential PFAS sources right next to our environment. So if you think of like the common potential sources, like a manufacturing site that may be making PFAS or using PFAS as part of its manufacturing process, that potential PFAS waste is then dumped into the environment in effluent, or maybe it's emitted in the air. And these PFAS are so strong that most combustion, most heating methods don't actually break them down. They just volatilize them and put them in the air, which eventually go down in rainwater somewhere else. So they are a problem not just where it's actually a source, but also a problem elsewhere. We also have farms that use biosolids. Biosolids are full of PFAS. And they've been showing that in certain crops, the biosolids that release the PFAS will actually incorporate themselves into crops. Landfills, which are passive receivers of our waste. Our waste is everything we use that has PFAS in it. And landfills have become a depot of PFAS. All this waste goes, all the PFAS gets put to a landfill. What I've learned, and something I'll talk about later, is that not everything that goes to a landfill stays at a landfill. So, for example, biosolids from wastewater treatment plants are then often sold to farms. Landfill leachate, which is the garbage juice that's generated from all this waste, can be taken to a wastewater treatment plant for processing. Unfortunately, wastewater treatment plants only remove about 10% of PFAS that enters their processing facilities. All that then gets dumped into the water. In Florida, I have some places where they do deep well injection of landfill leachate. So they actually dump it straight into the earth. And Florida, as an island, obviously you can imagine that's not really a great idea as well. And again, we mentioned that PFAS can come into our environment through the use of aqueous film forming foams at airports. And why is this important? Well, in Florida especially, we get a lot of our drinking water from a lot of these water sources, right? The Florida aquifer, groundwater, and so forth. And if you start polluting those water systems with PFAS, we're going to start seeing it in our drinking water. And if you look up in Google and you type in PFAS and drinking water, you'll see a whole bunch of communities that have issues with PFAS in their drinking water. And it's most likely because of some source nearby. In fact, we just published a paper that showed that the PFAS that comes through Florida Springs, especially in the vents, has PFAS concentrations that are pretty high. So these things move around within our waterways pretty easily. So once we get exposed to PFAS, what happens? So some of the exposure routes would be obviously our water. Food is becoming a much higher PFAS source. So meats, poultry, and beef products have PFAS in them. Our indoor environment, dust, also has a lot of PFAS. We'll talk about that a little bit later. Once we get exposed to PFAS, they're not like most contaminants that bioaccumulate in your fat tissue, right? These actually bind to proteins in your blood. So they bioaccumulate in your blood and blood-rich tissues. And unfortunately, they're not readily metabolized. So they stay in your body for a really long time. And what's really scary is they cross the placental and milk barriers. So during developmental stages, PFAS can get into these developing organisms and cause havoc. This is just one of many different types of figures you can find on the internet. Shows all the things that PFAS have been associated with health-wise. I chose this one because it has a lot to do with development, which is something I'm very interested in my research group. Things that always caught my attention was the ability to decrease your response to vaccines. You know, obviously with COVID, that was obviously a big deal. But the list of things that PFAS are associated with is continuing to grow. Cancer, neuronal development, changes in lipid concentrations, the list keeps growing. And what's really scary is that everybody in this room has PFAS in their body. So it's not something that you can get away with, and that is going to constantly keep growing. So because this is an issue, and this is an issue that we can't really stop, we're obviously not going to stop buying products, and we're not going to stop throwing them away. EPA has started to become involved. And I'm not going to talk about everything on this screen because there's a lot here and I don't have the time to go over it. I'd be happy to talk with anybody afterwards about more of these things. But EPA started to get involved in 2009. It took quite a while before they actually started impacting regulations. So just this past year in 2024, they enacted these maximum contaminant levels in drinking water. And there's only maximum contaminant levels for two PFAS, PFOA and PFOS, and that's 4 parts per trillion. Now, part per trillion is basically equivalent to a drop of water in an Olympic-sized swimming pool. So it's very, very small concentration. And with this enacted contaminant level, the goal is, assuming this continues with the regulation that's been passed, is within 5 years, all drinking water has to be below 4 parts per trillion for these two compounds. So the water you turn on your tap, the levels have to be below 4 parts per trillion, otherwise someone's in trouble. And so that's great, but again, these are 2 out of 15,000 potential PFAS chemicals that we need to start paying attention to. So we're slowly at a snail's pace starting to become more concerned and putting things into place. So how do we actually measure PFAS? So I'm not going to get into too many chemical technique details, but I want to talk about a few, just so you have an idea of the things that we do and the techniques that we can apply to measuring the PFAS space. So the EPA and others that do regulatory analyses do what we call a targeted method. The targeted method means that we have a list of PFAS targets that we're trying to measure, and we only measure those, right? And this is great because it's very routine, robust, it's highly quantitative. But as you can imagine, there's still a lot of other space that we're not interrogating. So that's where non-targeted workflows come in. This is what allows us to look at some of the unknown space to see if there's other compounds that are potentially more concentrated than the ones we're already measuring. And the hope is that once we start identifying these new ones, we use that to feed into including those into our targeted workflows. But as you can imagine, the more we learn about PFAS, the more we learn the space is wide, right? There's a lot of different parts of PFAS landscape that we needed to interrogate. For example, the super volatile ones that go into the air, that enter indoor spaces. There's different types of chemistries that we're not capturing with the current techniques. For example, like ultra short ones. And then there's a technique called total fluorine, which we'll talk about a little bit later, that we're starting to implement that will allow us to actually determine how big the PFAS landscape is. So just again, briefly, I'm going to talk about how the targeted workflows work. So again, I told you that before that these are what EPA uses in all regulatory laboratories. And that's because they're all validated by standards. So every one of the chemicals that we're measuring, we buy a standard, we optimize it in our mass spectrometer, and we get the best settings to get the best sensitivity. And we quantify everything by spiking in isotopically labeled standards, analogs of the things we're measuring. And we can detect to low parts per trillion. And so basically how this works is this is a fragmentation profile of PFOA, one of the legacy PFAS. And you can see here we have its mass at 413. And we have a bunch of things that are to the left of those. Those are fragments. So we take the parent mass of 413, we add energy, break apart, and we look at the pieces, kind of like a puzzle. You have an intact puzzle, you break it into pieces, and those pieces of the puzzle correspond to something as part of the entire puzzle, right? And so with this mass spectrometer, we have 3 quadruples. You don't really need to know what a quadruple is, other than the fact that it's a mass filter, okay? So what we can do, and this is why this provides us the greatest selectivity and sensitivity, is we can tell the instrument that we want to look for only mass 413 if we want to look for PFOA. Everything else that has a different mass is discarded. So we've gotten rid of all the things in the sample that don't have a mass 413. In the second quadrupole, we have a collision cell. So we break apart that mass 413 into its fragments. And then the third quadrupole, we measure all the fragments. So all the things that were fragmented from the original 413, we measure. And this is what we see in our profile here. So we break apart 413, we get these two distinct fragments that have to deal with certain parts of how its structure is fragmented. And we can use what we call transition. So we can tell the instrument to look for 413 in turn breaking down into 169. And we have 413 breaking down to 369. So this is how we take thousands of things that are in a sample and focus in on the specific details of certain chemicals. And we can do this for all the individual PFAS. So the rest of the talk, I'm gonna bring up a few applications that we have in our lab that I think are really interesting, and in 4 different categories. And the first one is gonna be environmental. So really interested in water, and PFAS readily are found in water, they move in water, and in Florida, water is a very important resource. We use it for drinking, obviously we fish, and there's recreation, and there's very little data if you actually look into how much water is in Florida and how much we actually know about it. And so I wanna try to fix this. So we came up with the idea of crowdsourcing sample collection. So we reached out to students at the University of Florida, and we asked them if they'd be interested in helping us collect samples across the state. Now, not that there's any benefit to having COVID, but this project started during COVID, and all those students who were interested in participating were all back at home all over the state. So it really allowed us to start trying to have a wider coverage of our sample collection. Eventually, we got citizen scientists and other citizen organizations involved as well. And overall, we had 130 undergraduates help us collect samples and 75 citizens and organizations. And what we did is we have a sampling kit. We have a YouTube video that showed everybody how to collect the samples properly. And in this project, we collected over 2,000 sites. So it was a very productive project. The map on the left is actually showing the some PFAS. So we measured 92 different PFAS in our workflow, and 33 were detected across all the samples. And so what some PFAS means is we added all of the concentrations for all the PFAS that were detected at each site. And you can see all these dots. The red ones constitute areas that have higher concentrations, and green is lower concentration. So immediately you can kind of see that there's some sites that, especially Tampa Bay, have a really high concentration of PFAS. One site in particular was over 3,000 parts per trillion. Remember, 4 parts per trillion is the drinking water MCL, and this was near the Fort Lauderdale airport, and the mean was 29 ppt. So what's really great about having all that data and all those sites is that we can generate heat maps. And heat maps are useful because they allow us to predict what levels would be in areas where we necessarily don't have a sample. And so how heat maps are generated is that we have each site has a concentration, right? And we have a coordinate, and we have coordinates and concentration for all the sites around it. And understanding those relationships allow us to generate these nice heat maps. And again, this is a heat map of the sum of PFAS. We can also look at individual PFAS. So this is a heat map of just PFOA. And you can see that a lot of things are similar, although there are some areas where PFOA may be higher than found for other PFAS in the sum PFAS. So it allowed us to try to know maybe where there are potential certain types of sources that could be investigated further. And oh, by the way, we had over 900 sites for just PFOA that were above the 4 parts per trillion. So a lot of water was very contaminated with PFOA. So once we had these heat maps, one of the things we want to do is try to provide some context. Now, going to this, I thought, well, we have a lot of military bases and airports that are really close to water, they're coastal. I thought maybe the use of AFFF was the main reason we were seeing these high levels. But then we stumbled upon this website, which is very hard to find, but it's run by the Florida Department of Environmental Protection. And what they do is report all the chemical spills, self-reported, that have occurred in the state of Florida. And so if we look at the time period from when it started in 2017 to when we ended our sampling project, there were over 7,000 spills that occurred in the state of Florida. And these spills range from different categories of raw sewage to treated, untreated wastewater. sludge, and other miscellaneous things like gasoline and other chemicals. In total, there's 1.4 billion gallons of stuff that was dumped in the water. And this is self-reported, so this is an estimate. So it's likely this is probably underestimating. If you were to take all those locations and plot them, this is what it would look like. So these are the spots of where all those spills occurred. And 22% of the reports in this Excel didn't have any gallon information. So there are a lot of other spills that are noted here, but actually don't have a quantity. So again, most of this is highly underreported. What struck me though is when I started comparing the spill locations with our heat map, they look very similar. So again, AFFF usage is probably a significant source of PFAS pollution, but so are these spills. Now, spills could be happening for a lot of different reasons. We have hurricanes, right? And hurricanes often flood a lot of the places where these spill components are treated, right? A lot of times sewage can overflow and so forth. But a lot of these are just routine spills and millions of gallons estimated. But you can see here that there's a very stark similarity between the spill locations and where we see highest levels of PFAS. So the second part of this was actually to compare not just the site of where these spills are happening, but the quantity. So I have all the different categories: raw sewage and treated, untreated wastewater and biosolid sludge, but I'm only going to show one, and this is raw sewage. So this is the Tampa Bay, Pinellas, Hillsborough County map. These are the sites of spills. And this is the quantity of spills that occurred in this area. So again, this is one of the hot spots for PFAS. And we see upwards of 6 million gallons of just raw sewage has been dumped in this area over like a 5-year time period. So this is a pretty significant source of PFAS. To put in more context, we like to do back of the napkin calculations, right? Again, these are estimates. But if we look at the amount of gallons on the top two lines, either from the 5 years we studied or up into the current date, you can see there's hundreds of millions of gallons of each of these types put into our waterways. If we look at how much potential PFOA is released in quantities, in grams, we can take what the average concentration is reported in literature for those 3 different types: influent, effluent, and wet sludge, and multiply that by the gallons. We get these. This is how many grams we get incorporated into our environment every year. So, for example, the influent, it's close to 19 grams of PFOA release over that time period. You could do the same thing with PFOS. And what's interesting about PFOS is that even though wet sludge is a very small constituent compared to the influent effluent, it has pretty much a similar concentration of how much is released. It's because PFAS is in high concentrations, as you can note here, in wet sludge. Also, just want to point out too, I mentioned before that wastewater treatment plants don't effectively remove PFAS, and this highlights this, right? So our effluent is about 90% of our influent. So we only remove about 10% of PFAS once it goes through these treatment facilities. So one of the things we're working on now is taking our target method, but also implementing other PFAS measurement strategies. And the 2 sites that we're doing these new strategies are Tampa Bay, because we know it's a hot spot for PFAS. We're also working in the Mississippi coastline, which has a lot of PFAS sources, but nobody's really done any measurements on yet. And the 2 different techniques we're employing are what we call total fluorine analysis. And we're looking at 2 different methods for this. PIGY, which is particle-induced gamma ray emission, and combustion ion chromatography, CIC, for total fluorine. And then we're also doing this high-resolution mass spectrometry non-targeted analysis. I'll briefly talk about what those are in a second. We're also looking at new matrices for us understanding the impact of PFAS in our environment, sediment cores, and foam, which I'll also talk briefly as well. So what is total fluorine? So if I were to take our targeted method, let's say of our 40 compounds that we're measuring in our EPA method, we cut off all the fluorines and measure the concentration of fluorine. That would be the total fluorine of those things we're measuring in the targeted method. But if we were to take an entire sample, let's say we have a bag of water, and we cut off everything, all the fluorines off everything that's in that sample, we would get the maximum total fluorine concentration, right? If we compare that concentration to the same concentration of fluorine from our targeted method, we can understand how much space is left to be interrogated that we're not currently interrogating with our targeted approach. And this is useful, right? Because it sets the upper boundary of how much more is left in the PFAS space. And what really got me interested in this was a paper that was published last year from South Carolina. And basically in this paper, this is by Susan Richardson, they looked at both the targeted method, which is the blue lines, and the total organic fluorine, which is the orange lines. You can see that the blue lines only estimate a very small percentage of the total fluorine that's in these samples. So this got our attention that, hey, maybe we need to start looking at whether or not what we're measuring is actually a big part of the total PFAS landscape. So once we establish that some sites have a big disparity between our targeted method and our total fluorine, well, how do we get to that unknown space? And that's where we use non-targeted PFAS workflows. And so this is our instrument. It's an Orbitrap. It's a high resolution mass spectrometer. And the way I like to describe it is you have a light and a dark space. The light is what we measure with the targeted method, right? But we need to figure out what's in all this dark space that's around here, the things that we're not currently seeing. And so this will allow us to start trying to identify what some of the things are that make up this extra space between our targeted and our total fluorine analysis. So what I have here is two things. On the top, I have, let's see if this plays. Let's see. Nope, hold on a second. You can see I had to add a video. Where do I click on it? Okay, thank you. So the top you see total ion chromatogram. And basically the size of these peaks just says how much intensity of things that were measured in the mass spectrometer. And the mass spectrometer is scanning at each one of these time points. What you see below are all the masses of all the compounds between a mass range of 200 and 1200 that show up at any one of those times. And you can see as you move across, there's a lot of different molecular signatures that are there that we can interrogate and figure out what those are. And we've developed software that'll actually go in, look at all these masses and identify putatively what all these unknown PFAS are. So in any sample, depending on what it is, we could measure 50 to a couple hundred unique PFAS that we think are putatively a certain type of chemical group within the PFAS landscape. And then we can then buy those standards if we can get them, and we can actually do analysis to see if they're really PFAS and incorporate them into our targeted method. So we're trying to interrogate that unknown space. The other thing that we're doing right now is looking at different matrices. So this is my student, Isabella, and sorry. Okay. Oh, there you go. Oh, thanks. All right. So this is my student, Isabella, and what she's doing is she's taking a sediment core. So a lot of the work we've been doing in the past, we take the surface of the sediment and we get our concentrations. Well, let's say you want to do a retrospective analysis. So you want to look at what levels of PFAS occurred over time. And so what we started doing is sediment coring. So you need to know the sedimentation rate of where you're analyzing. In this case, in Mississippi, we knew that the first 5 centimeters were within 8 years. The next set of 5 centimeters were 8 to 20 years, and then past that, over 20 years. So if we have a site that we're interested in trying to study, let's say at Keymore's plant, which is in Mississippi coastline, that's where a company that makes PFAS, we can actually grab these sediment cores, which Isabella is doing right now, and she puts this over a PVC pipe, kind of like a push pop, and then all the water is extruded, and then we actually cut the individual layers. And then we can do a PFAS analysis of that. So we can actually see over time whether or not PFAS levels have gone down or have gone up. I'll just let this video go for a second here. The water is removed, and then we measure 5 centimeters. We take off the 5 centimeters, and then we put that in the Ziploc, and then we dry that sample and then do our PFAS analysis. And we're also going to be doing total fluorine analysis on this as well, and then take the next 5 centimeters and so forth. So the other matrices that we're doing that's really cool is foam. So you might have witnessed foam on coastlines. It's a naturally occurring thing. What we found, though, is that foam is chock full of PFAS. So on all the sites where we collected foam and an adjacent water sample, the foam was anywhere between 30 to 1,000 times higher in concentration. And in some sites, we were measuring in upwards of 100 ppb in the foam. So really concentrated, don't play in the foam. And this is actually a picture in Florida of an area near a hurricane that generated tons of foam. It kind of looks like somebody dumped a bunch of laundry detergent or something, and it created this big foam party, but it's not, it's naturally occurring foam and likely to have a lot of PFAS in it. That's just me collecting foam. So also, we do a lot of work with biota. So once these things release into the environment, we try to track their presence through various food webs. And one of the things that we developed in a lab was measuring PFAS in blood. So I mentioned before that PFAS bioaccumulate in blood. So it's a great matrix to non-lethally test how much is in an organism. And we basically do a protein crash. So basically you add methanol or acetonitrile, you crash out the proteins and you take the top layer. We actually adapted this to a 96-well plate format. So now we're starting to do more high throughput analyses. And what's really cool is they've actually started to develop these self-collection kits for humans where you can actually pay, they'll send you this Mitra test kit. Basically, you use a lancet to create a drop of blood. You put that blood on these tips right here, you send it back and they do PFAS testing. And we're really interested in trying to apply this technology to environmental studies as well. So one of the first environmental studies we did was looking at alligators, which I know many of you are very familiar with. And this is a study we did at the Merritt Island National Wildlife Refuge near the Kennedy Space Center. And here we looked at almost 230 alligators over one period of time. And this is 10 males and 10 females each month over this time period. What you're looking at is a heat map of PFOS. All the dots are actually individual alligators, and then the colors represent the concentration range. So the darker, more concentrated. And some of the highest sites that we found were near where they do their AFFF training exercises. So they may not have fires all the time at these facilities, but they do a lot of training. So they use these film ring foams. Also near the launching facility was another place we had high levels too. And one of the highest animals we had was over 2,000 nanograms per gram, which is really high. Then we also looked at PFOA, and we see two sites that were really high. One is where they do the AFFF training exercises, but another site was actually where they had an old artillery range. And it turns out that bullets used to be lined with PFOA. So one of our hypotheses is that's the reason why we're measuring PFOA at high levels in this area. Since that study, we have over 1,000 alligator plasma samples collected at NASA, and 50 of them have been recaptured with a 5-period gap. And so we have an ongoing study. We're looking at individuals and how the levels change over time. And why is this significant? Because I mentioned before that they started to phase out a lot of the legacy PFAS in the early 2000s. But in coincidence with that, they also started introducing new PFAS, these alternative PFAS chemicals that are now being used in replacement of the legacy. So we're really interested to see whether or not in the wildlife, like in humans, those legacy ones are going down and other ones are going up, the new replacements. And we have never really done that before in a wildlife study because we never had access to these type of samples. We're also doing a lot of monitoring of PFAS in alligators across the state, hoping to match that with the water concentrations we have. And then we have 5 sites where we have a 20-year gap between collections. So we have samples from the late 90s, and we have more contemporary samples that we're going to compare to see, again, if those levels have changed over time. We also had a study where we looked at PFAS in manatees. So most of the routes of exposure that you have by biomodification occur through carnivorous routes. And so we want to look at a herbivore to see whether or not they also had high levels. And in the Indian River Lagoon, they actually had really high levels. And they're almost predator-like levels. And so we were wondering why this was the case. And it turns out they eat a lot of seagrass. And so we did a follow-up study where we collected seagrass near a lot of these suspected sources. And it turns out that near the Kennedy Space Center, near the Patrick Air Force Base, and near a wastewater treatment plant, we had the highest concentrations of PFAS in the seagrass and macroalgae that were present in those locations. So we propose that this is a major source of PFAS for the manatees. We're also working with sharks. And so we did a study looking at shark concentrations of PFAS in plasma. This is all along the Atlantic. And this is just a quick piece of data between the 4 different species. And what we learned is that bonnethead sharks had the highest concentrations compared to other sharks. And one of the main reasons that is because they're benthic. So we've been learning with a lot of things that we're looking at with species that those that live on the bottom of water actually have higher levels in comparison to other organisms. And that's because PFAS bind to particulates that then sink to the bottom. And if you're feeding or foraging through the bottom sediment, you're going to start increasing your concentration levels. So this is something we see in sharks, but other animals as well. Things like catfish and stingrays are something we also see very high levels in. We've been continuing to work with sharks, and one study that we're in the process of submitting, we're looking at maternal transfer. So working with Florida Fish and Wildlife, when they have sharks that are stranded, they let us know that they have potential samples. In this case, we had pregnant sharks that were found stranded and dead, that they then provided tissues for us. So we were able to compare the liver samples and muscle samples from the mother to the embryos. And you can see here the bonnethead mother liver samples were really high. So again, sticking with that benthic benthic source of PFAS contamination. But you can also see that a lot of the embryos do have PFAS in their liver. And in the muscle, actually, they're pretty comparable to what the mother has. So PFAS are maternally transferred to these offspring. And again, we don't have a lot of data because these are very hard samples to get, but we did notice that the one shark that was aplacental viviparous had the lowest PFAS levels. So we think that the placenta is actually one of the major sources or pathways for getting exposure to the embryos. So if you don't have a placenta, then that may be that you have less PFAS in that system. So again, not a lot of samples, but something very interesting that we found. We're also working with other sentinels like pythons. So as you might be aware, Burmese pythons are invasive species in Florida. They're taking over. And so they're constantly calling these animals. And so we're working with people down there to actually provide us with samples when they do this. Then we can actually measure the PFAS levels in both the left side and right side of the Everglades, where people are living, but obviously these animals track for both areas. What really makes them nice is that they're invasive, so we don't have to worry about harming something that's not already being harmed. They're dietary generalists, so they eat everything, and they're apex predators, so they're a good potential sentinel, and they both live in the aquatic and terrestrial environments. Overall, we found 9 PFAS. And again, something we're seeing with a lot of organisms that we're testing is that there's a difference between the profiles between males and females. So whether or not that's offloading through reproduction or some other mechanism, that's something our lab is really interested in interrogating further. Something that I also really like doing is coming up with new unique sentinels. So I hate ticks. So this was a really easy project for me to take control of. And what we wanted to do was compare ticks found in contaminated areas. So Sweetwater is actually something that's in Gainesville. It's a man-made stormwater wetlands treatment facility. And we had much higher concentrations of PFAS in ticks at that facility. Now, ticks consume blood, right? And PFAS are in blood. So what we are proposing, and what we want to continue to keep working on, is that ticks are ecosystem-wide sentinels of PFAS burden. So instead of going out and sampling all the organisms within an environment, you can let the tick do it for you, right? And you can sample them and then figure out what the burden is in that area. Something, of course, we also want to do with leeches. That's something we want to start working on at some point. We also do a lot of work with fish as well. So we have a study ongoing where we've sampled close to 2,400 fish muscle samples from a lot of different species, working with FWC and charter boats and seafood markets. We also have an ongoing freshwater study as well. The goal of this is to have data-driven health advisory level for consumption. So right now, there isn't any in Florida. So there's not information about which species are the worst to consume if you want to reduce your PFAS burden. And that's what we're hoping to do with this study. All right, so the last 2 parts I'm going to talk about just briefly is waste streams in our indoor environment. So you might say, well, why should I care about these things, right? And I had that same thought when I first started. But what you really realize is that our waste streams are intimately related to our environment. So let's say we have a PFAS material that gets thrown away in a wastewater treatment plant. Well, we already talked about that PFAS are not removed when the wastewater treatment plants actually process. So that goes right into our surface water, which then can be then used for drinking or get into aquatic life like fish that we then consume. But let's say we also have biosolids. Well, biosolids that are generated from wastewater treatment plants those can be sold like they do in Florida for surface application for crops, which then get to our food, which then we eat as well. If they dispose of the biosolids into the landfill, that's great, but we already talked about landfills not really being a constrained environment. They actually take their leachate, process it through a wastewater treatment plant that then goes to the surface water and so forth. So this cycle of constantly producing PFAS waste ultimately ends up in ways that we might be exposed. So one of the things we work on in our lab is methods to try to remove PFAS from these matrices. So on the left is concentrations of PFAS in landfill leachate. You can see a lot of these are in the thousands to multiple thousands of PPT. So leachate is a very concentrated matrix. Unfortunately, all the techniques we have to destroy PFAS are high energy and expensive. So if you have gallons and gallons and gallons of landfill leachate, that's not going to really be effective. So we came up with a technique that's very simple of foaming the landfill leachate. So if you were to basically put an aerator in a big vat of landfill leachate, you create this frothy foam, you remove that foam then coalesces into a liquid. We've now size reduced our landfill leachate from 700 mils to 100 mils. So we've concentrated all that PFAS into a smaller volume. And it turns out that when you do the foaming, it actually moves from most of them upwards of 80 to 90% of the PFAS. So it's a very effective method, which we've actually in the process of patenting. And we're hoping this could be something that's applied to a lot of treatment facilities. Another thing that we're really interested in is tracking what happens to what we call precursors. So I mentioned before that we have these legacy PFAS that are things that have been around for a long time. They're in everything. As we start fading those out, all these new alternatives are starting to be introduced, right? Some of them we don't even know about, right? They're proprietary. And these PFAS precursors, why they're so important to study is that they're made up in the chemical structure wise of basically pieces that have the legacy compounds attached to them. So over time, they can produce the legacy compounds when they break down. So we may not be making these legacy compounds anymore, but indirectly we're making them as these precursors break down. So what we want to do is understand what happens to their fate once they're going through the landfill process. So we have our waste that we throw at our house. We put in a trash can, a dump truck, garbage truck will come by, and I'll pick up your trash, right? And this will go to a transfer station. And a transfer station is just a place for all the trash to be put together that's then taken in bigger trucks to the landfill. And when you compare the level of terminal species, the PFAS, which are green, the landfill leachate has a lot of these, right? And then our fresh waste has much lower. We have a lot more precursors, right? So these are things that are used in paper products, food packaging, things we throw away that have all these new alternative PFAS. And so part of the study was to have our students collect leachate from the back of a truck, which is obviously very gross. And we did the analysis, and if we compare the bottom two, which is residential waste and commercial waste, to a landfill leachate, you can see the yellow bars, which are precursors, are really high. But through the course of the landfilling process, those are all bricked down to a smaller concentration. What ends up happening is now we have a lot more terminal species. So all those big precursors now are breaking down into all these terminal species. And why is that important? Well, we're generating more terminal species that can be then taken to a wastewater treatment plant, released into our waterways. So the cycle continues. We're also really interested in beneficial reuse materials. So a lot of things like, let's say concrete from roads. When roads are remade, all the old concrete gets taken and reused as something else. Same thing with street sweeping. So when the street sweeper goes by, collects all the debris, that gets taken and used in other materials. We wanted to see whether or not those materials had PFAS, and we should be concerned about how they're reused. And so we did a study looking at street sweepings across the city of Gainesville. And it turns out the highest site, which is this huge bar right here, is near a Superfund site. So this is a site that likely used lots of PFAS for their activities, and we're constantly measuring it in the street sweepings around it. So just another thing that we have to pay attention to. If we look at street sweepings from different cities across the state of Florida, the green is short chain PFAS, the orange is long. You can see that even within different cities, we see different profiles. So there's lots of different uses that are going on in these cities. Not all cities are created the same. So this data is important for Gainesville, but it may not be applicable to other cities, and more work has to be done in this space. And very briefly, I just want to talk about composting. So one of my students, Alina, she's been working on composting for a long time. She did a first study We looked at whether or not food contact materials generate a lot of PFAS waste in composting, and the short answer is yes. They're actually the biggest source for PFAS in composting. So don't put anything that says compostable, but it's food contact materials in your composting if you do that at home, like the clamshells and other containers. This actually started pushing us another direction where now we're in the process of doing a US-wide compost study. We have close to 400 samples. We're measuring PFAS, microplastics, and heavy metals in various composts. So we're looking at different feedstocks, so things that people put into composting, different compost strategies, and also things like crops. Once we apply these compost to crops, which crops are the worst in terms of incorporating PFAS into them. This is something we have ongoing. The last few slides are going to talk about indoor environment. We have lots of work in this space, but I'm going to just talk about one thing that I think is kind of eye-opening, at least it was for me. In our indoor space, we have a whole bunch of potential PFAS sources. And this just covers a few, from cosmetics to food packaging, rugs, carpets, garments, furniture. This would be something you might find in your house, right? But there's plenty of studies that are showing there's these same things that are found in your childcare facilities and schools and in our workplaces, right? We have lots of different potential PFAS sources. And what I want to get to is that dust is a really useful composite matrix to assess what those indoor burdens are. So we did a study where we looked at whether or not we could use home air conditioning filters as a new matrix to assess how much we're exposed to in our living spaces, right? We put these in our houses to filter out dust and other air particulates. And we know PFAS bind to particulates. So in this case, we had homes that we tested their filters. We also had facilities on campus that basically filter out air through a large building. And the great thing about this is it's throwaway, right? We already throw these away, so we're not asking for anything new. And we can then assess our burden over time. So first question is, do we find PFAS in these matrices? And the answer is yes. So we found 27 PFAS in these air filters. And how much is in there? So mentioned before, parts per trillion is where we want to get to with water. We're measuring in the upwards of high parts per billion, even low parts per million PFAS in these dust air filters. So while we are concerned about drinking water, our indoor spaces are also an extremely important thing to start paying attention to. And just as an analytical aside, we tested all the new filters and didn't find any PFAS. So all the PFAS we're measuring is from that dust that's collected by the filter. We look at different PFAS groups across the campus and household filters. And what we noticed was that the household filters had a greater diversity of PFAS profiles, which makes sense, right? We all have different houses and different potential sources, and it had higher concentrations. But what we found was these DIPAPs, these polyfluoroalkyl phosphate esters, were the highest. And in some cases, they were constituted about 95% of the concentration of PFAS. And why are DIPAPs important? Well, they're another class of these alternative precursor species. And they're used in high quantity in paper products and other materials. And why are they bad? Because again, as I mentioned, these DIPAPs, in this case, they're made up of this middle group here, and then two PFHXAs attached, will eventually break down into PFHXA. So we may not be producing PFHXA anymore or using it in products, but indirectly we're making those through the destruction of these new materials. And one area that we're really interested in investigating is the effects of these on children. Obviously, children playing on the floor are exposed to dust at a high level, also pets. And another really important aspect to consider here is that, again, we are collecting all these filters, we're throwing them away. These are also new waste streams, right? It turns out we actually see PFAS in the air filters in cars too. So that's another paper that's coming out very soon. So just doing a back of the envelope calculation, if we have a 50,000 person city and they replace their filters 4 times a year, that's about 11 grams of PFAS that's added to a landfill every year from just this one source. So again, there's a lot of things that we need to study and understand, but this is another significant potential source for PFAS. And that's the end of my presentation. I like to include this slide because it's always important to ask for help. And even PIs need to ask for help sometimes. This is Hannes in South Africa holding my pants as I'm trying to sample this water. So I'd be happy to answer any questions you might have, and thank you for your time.
Before we go to questions, I wanted to a little context. In the spirit of this being the graduate student seminar series, I conveniently forgot to say that Miriam Boucher is the whole reason John Bowden's here today. So Miriam, Dr. Bowden is on Miriam's committee. She's been going to his lab for the last year or so to analyze her PFAS samples, and John's provided a lot of guidance for Miriam. So that's why he's here today. And it's not because of me, it's because of Miriam. Take it away.
Any questions? Yes.
Do you have any good news?
No, so I like to joke that I'm not very fond of parties, right? Because people ask what you do, and then you start talking about. I mean, I would say that you know, the government's starting to provide some initial regulation for these, which is good. But part of it is just educating people, right? People, you guys are obviously very well informed. Most of you know what PFAS are, but most of the time when I give these talks, people don't know what PFAS are, or how prevalent they are, and just about everything that they do or touch are exposed to. So unfortunately, no good news other than the fact that we're trying to fight the fight and provide that information for people to make educated decisions about what they buy and use. But unfortunately, these products are still being made. Most of the PFAS that we don't have standards for, the other 14,000 are proprietary, and they're not sharing the information with people like me to measure them. So a lot of progress still has to be made to get to a good place, unfortunately. Yes.
How and why South Africa?
Yeah, it's a good question. So Lou Gillette, who me and Thomas had worked for a long time, we started a project looking at this disease called pancreatitis in crocodilians there. And we went on a few projects, and we actually detected some biomarkers that allow us to detect the disease without killing the fish, which is something you couldn't do previously. And we developed some really great connections over there. And so I've actually been there like 6 times. And so each time we go and do a different project. So we've done projects looking at road dust, contaminants of road dust. We've done projects looking at water. We were in the process of publishing another paper looking at composting strategies in South Africa and emerging contaminants. So it's a really great experience to bring students. I brought several students over there. It's really good for them because here, sometimes it's not obvious what research does in terms of impact on society, but there it's very visceral. You can see exactly why you need to do the work there because everything's kind of out in the open. You know, things aren't hidden. You know, people are dumping trash right into the water that's destined to go use on a farm. So it's a very cool experience to do international research and kind of help students grow in understanding the impact of the research. And also they're there talking with other students from local universities and can, you know, there's a lot of technology transfer that occurs. So we do work in Japan and other countries as well. So it's a lot of fun to do international research. Yes.
Framing this local.
Yep.
I volunteer and I'm on the PD River Basin Council, which is looking at the whole PD river. I don't even know if you know what the PD River is, but our local water. And 3 local water companies, the city of Georgetown, Georgetown City Water, Horry County, and neighboring county, are suing some businesses because of the chemicals involved. And the answer we're getting is EPA, like they aren't going to enforce anything until 29. So we got all these chemicals, but here's a room full of people who seem awful healthy. So what's going to happen if we keep ingesting these, drinking them in our water, and it doesn't appear that anybody's suffered any consequences? Yeah.
Well, I think one of the things that is a misconception is there's lots of chemicals that if you ingest it, you die right away, right? And you know those are bad, right? There's a lot of chemicals that take time, and PFAS, from once you're in utero, you're exposed to these chemicals, right? And it's really difficult to quantify the impact that has on your health as you grow and develop, right? But what I'll say is that if you look at health implications in lab models and association with people's health. It's pretty dramatic in terms of how bad these things are for our health. Now, you may not suffer, but those PFAS that you have in your body might change your DNA, and maybe your offspring are going to have problems, and their offspring are going to have problems. So there's a lot of different ways that these things can potentially have a negative impact that you might not notice right now. But again, that's really difficult. That's where a lot of these companies are sitting behind in terms of not necessarily paying out for a lot of these issues, is because it's really hard to link exactly what's happening to a person from exposure